maybe we can go a little bit back in time. I was a physics undergrad in South Korea, Seoul National University. And to me back then, proteins meant just one thing. It meant something that you eat. There are three nutritional ingredients: proteins and carbohydrates, and what's the third one? Fat. Okay. So when I learned toward the end of my PhD training in the US that proteins are actually molecular players that perform important functions in the living cells, it was a complete surprise. But now I know that proteins are responsible for almost everything that we do. When you think, when you dream, when you sense mechanical forces with your fingertips, when you smell, when you hear, it's the proteins that are actually doing all the important functions. And they are very small, and they function more or less autonomously, so you don't have to tell them exactly what to do at every moment. And so to understand how they function, we need a technology that can measure them while they are functioning, when they are not actually fully synchronized. And that's the reason why it's important to study proteins one molecule at a time. Let's say you go to a college campus, there are a lot of college students running around doing their own things, and it's hard to synchronize them, right? They are not soldiers in the army. They are not marching in synchrony. So if you average their movements over 1,000 college students, you will not be able to figure out what is going on. You have to look at individual students one at a time, to understand their behavior, why they do it, how they do it. So let me give you an example. So when you think, actually, okay, I need to move my muscle, right? You need to send your signal from your brain to your muscle. And therefore, your neurons are very, very long. The nerves are very, very long. But to communicate between your brain and your muscle junctions, you also have to transport important protein molecules or important RNA molecules down the nerves, and this can take a long time. So if you rely on random movements, such as Brownian motion, it may take days before you can go from your brain to the nerve's end. Therefore, you need to have what's called motor proteins. These are proteins that can move along a cellular highway using ATP as a fuel, just like automobiles use gas as a fuel to move along the highway. They use ATP to move along the track. Now, if you want to understand how they actually function and carry large cargoes over long distances, we need to be able to measure individual motor proteins movements in real time. And this has become possible in the last 20 years or so. It's due to the advances in light microscopy, advances in mechanical manipulation tools, and advances in preparing neurons and samples in a manner that is fully compatible with advanced biophysical studies. And now we know far more about how they do their job than before. And in fact, we can go and look at what happens if there's a mutation in these motor proteins that are known to cause serious genetic diseases in brain disorders and muscular disorders and so on. So many people like to call these proteins nanomachines. There are two reasons. First, they are incredibly, almost unconceivably small, right? So they are nanomachines because they are just about few nanometers across. About the smallest object you can see with your bare eyes is the human hair. That's about 80,000 nanometers across. So you can imagine how small a protein can be, right? So if you scale up your body to the size of the Earth, let's say I'm, the size of the Earth, then your single cell in your body will be about the size of the city of Madrid, and a single protein in that cell will be about the size of a soccer player, okay? So, but the modern technologies can actually visualize in real time the movements and skills of this soccer player from a distance as far as on the surface of the moon. So microscopy, tools now, you can use them to record in great details, how your favorite soccer players, runs and take sharp turns and deliver a winning goal in a soccer match. That's pretty amazing. We are studying protein motor molecule that it was known to move on the DNA. So you're doing single molecule imaging experiments, and in fact, there we are using a technology called FRED, but this is a reporter that changes color depending on how far away two molecules are from each other. And from this reporter, we were able to saw gradual movements of one protein along the DNA, like that, which was fine. But we found that once it gets to the end of the track, instead of coming off the track, it reappeared at the beginning of the track, and then went through the same motion, and then dissociated, reappeared, and did this many, many times. Based on these early findings and additional experimentation, we are able to show that this scanning movement, or we call patrolling movement, is used so that the protein can patrol the DNA street and keep it clear of proteins that can otherwise accumulate and cause DNA damage and potentially leading to cancer. So in this sense, these repetitive patrolling activities of the protein that we discovered is important in keeping the threat of cancer at bay. And now we understand at least one reason why when this protein is absent or defective, the cancer frequency dramatically increases. So then, based on the understanding, one may be able to come up with intervention or therapy. So that really I'm not qualified to even speculate, because my topic is highly fundamental basic science research. However, the one area where this could really help is the following. So we have technologies that can visualize these protein movements that we believe are important in keeping the threat of cancer at bay. And so then if you combine our measurement technologies with screening, so you can have potential 10,000 cancer drug compounds, and you can add them one at a time and then see what they do, right, to the single molecule activities we are measuring, in our studies. That may be the most direct way of actually screening compounds, because we'll be measuring their effects on the important protein activities. Usually, when you drug cancer compounds, when you screen potential cancer drugs, you give them to the cells, and then there are many, many steps before cells can show you a response, right? And you don't know exactly why the compound worked or not, but if you are using our kind of measurement technology at the molecular level, maybe you can get the most direct method of screening for compounds that can be potentially active. In my lecture, I will present unpublished data that's really, really exciting. So we actually found a method to use light to tell the CRISPR proteins to cut the DNA exactly when we want them to do it and where we want them to do. So we now have a full control of in space and time where CRISPR proteins can cut the DNA. Just step back, so CRISPR technology has really revolutionary biology, and also many important applications are arising in agriculture, in human disease therapies, and so on. The reason is that in the past, if you wanted to repair DNA sequence mutations that can be causative for diseases, there was no really a good tool for doing that. But with CRISPR-Cas9 technology, you can actually go into the cell and cut out a defective gene and replace it with a functional copy, on demand. And it's possible that in the next 5-10 years, serious human genetic diseases can be actually cured using the CRISPR-Cas9 technology. But however, there is still a major problem. It's called the off-target effect. You want to go and fix the DNA exactly where there's a problem, but our human genome is very, very large. There are 3 billion base pairs of DNA, which is about 1,000 times larger than the genome size of bacteria. And CRISPR systems were developed in bacteria as an immune system against virus infection. So when CRISPR evolved in nature, there was no pressure to really optimize it for the billion base pair size genome. It was good enough to function in the million base pair size genome. So when you're looking at these large genomes in mammalian cells, that's where we can really suffer from off-target effects. And so if you want to cut here, but you can also generate cuts elsewhere, then this can lead to unpredictable consequences. Now, your question was, what can you do about this? So one thing that we have done, already published some papers on that, we use single molecule technologies to see how sequence mismatches, the off-target effects are caused by the DNA sites that have imperfect sequence matches, but that's still getting cut by the CRISPR-Cas9 systems. And we actually were able to show that this is because CRISPR-Cas systems can still unwind the DNA strands quite effectively, even when there is an imperfect match. And we then show that if you engineer proteins to reduce this DNA unwinding activity, you can actually improve the accuracy in targeting the DNA. So that's one example. But based on our understanding of how CRISPR-Cas9 systems go and bind and then unwind the DNA and then cut to start genome editing, as I will describe tomorrow, we've discovered really exciting and powerful technology that can control the CRISPR activity with great precision. Yes, I think this is really a deep question that we should all think about carefully, right? It's because the technology can potentially change the definition of what a human being is, right? And I believe this is essentially a sensitive question in Europe because I know that GMO products are not even allowed in the marketplace here, right? So I think the off-target effects and safety issues these are technological issues that will be eventually resolved, probably sooner than later. So the next question is that what should be allowed and what should not be allowed. And that has to be decided collectively by, I think, all of the nations, on this planet. And regardless of what is allowed and what is not allowed, whatever is allowed should be accessible to everyone, right? So that we can maximize benefits to the public. So I think it's a really difficult question. So in vitro fertilization was controversial at the beginning. But once people start seeing the joy of new parents, then it became totally acceptable. So it may take maybe just a few examples where CRISPR technology can basically solve a problem that could not be solved otherwise. So I think there'll be less controversy about curing diseases of people who are already born, right? So, but really, whether you should be allowed to change the genes before the birth, I don't think we want to do that personally, but we may get there.